Hey everyone, welcome back to my office. Today we are going to talk about the cell cycle and mitosis. Some of this might be review from previous classes, but it's good to know for what we're going to cover going forward. And it's kind of interesting too. So, first, why are cells so small? Four main reasons you should know, all of which we touched on a bit last week. One of them is the surface area to volume ratio. And that's basically the idea that as a cell gets bigger, the surface area increases, the volume increases, but the volume increases more quickly. So this red line is the volume, blue line is the surface area. Surface area is the membrane, obviously on the outside of the cell, and that just can't increase quickly enough as the cell gets bigger to keep up with the volume, because obviously bigger volume, bigger cell has to transport more things across the membrane, and it can only bring them in and out across the surface area, and the membrane or the surface area just doesn't increase as quickly. That's true for any three-dimensional shape. As you take the same shape and you just expand it, volume increases more quickly than surface area. Some other reasons we didn't talk about in class as much. One of those is DNA overload. And if you imagine a cell getting bigger and bigger and bigger, well, if it's a single cell, it still only has one nucleus. So if that cell gets bigger, 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 eventually you have one nucleus that's trying to run this gigantic cell, and it just can't keep up. It can't make mRNA quick enough to keep up with the demands of a bigger and bigger cell. Three is the idea of complexity and specialization. So this is more on an organism level, and it's the idea that it's easier to build complex structures out of smaller pieces. So one of the reasons that cells are so small is if you're making complex things like organs, eyes, brains, all those things, it's a lot easier to build it, complex details out of smaller pieces. I like to think of it like Legos. Both of these are houses made out of Legos. They're probably about the same size too. The one on the left, you can see, is made out of bigger pieces, and it's a whole lot simpler. It's just less complex. It's still a house. But the one on the right, also a house. It's also made of Legos, but they're smaller, so it's easier to build complex structures with more details out of smaller things. And if you're building something really complicated like a brain, you want that to be as detailed and complex as possible. So small cells is like building out of small pieces. Last idea is repair and replacement. If you ever get hurt or damaged or injured and some cells die, it's a whole lot easier to replace small cells than big cells. Like if your entire arm was made out of 10 cells and one of those cells died, you would just lose a tenth of your arm, and that would take a long time to replace. But cells are so small, you can lose 100 or 1,000 of them, and it's really not that big of a deal because they're easier to replace. Next, we're going to talk about the cell cycle, which is basically a way of describing the different phases of life that a cell goes through. It's often shown in this circular diagram here and broken up into these different phases. G1 is the first growth phase. So the cell might be growing, but this is kind of like the business as usual phase. So the cell might be growing, but it's performing its normal functioning as a liver cell, muscle cell, skin cell, you name it. Then when the cell enters S phase, S stands for synthesis. Synthesis means making something. The thing that is being synthesized is DNA. So during this phase, the DNA gets copied in preparation for cell division, which we'll get to in a minute. Then we get to G2 phase, where the copied DNA is checked for errors by different enzymes. And then finally, the cell goes into M phase, which stands for mitosis. That's what we're going to be talking about for most of the notes today. And that includes all these other pieces: prophase, metaphase, anaphase, telophase, finishing with cytokinesis, at which point you have two cells, and they would be back to G1, and the process would kind of keep going over and over again. There are multiple checkpoints at the end of each of these phases, and those are basically just to make sure that everything has happened correctly so far. There are lots of different chemical signals that will trigger a cell to go to the next phase or to start dividing, but there are also enzymes in the cell to make sure that everything has been done correctly That's why even when enzymes make mistakes when they're copying the DNA, almost every single time those mistakes or mutations are corrected by enzymes. If there are problems with the regulation, a lot of those can lead to uncontrolled cell division, and we call that cancer. So, the one thing that all different types of cancers have in common is they're all based on uncontrolled cell division. Three main types of cell division that you should be familiar with. First is binary fission. This is how really simple prokaryotic cells divide. Like we said earlier, they're a whole lot simpler. They basically have DNA, cytoplasm, ribosomes. It's a much simpler process where the DNA is copied and then it just splits in half. And like I said, that's how prokaryotic cells reproduce. And after binary fission is complete, you have two identical copies of the cell you started with. Mitosis is similar because you end up with two identical copies of the original cell, but it involves a few more intermediate steps, which we're going to get to in just a minute. More intermediate steps because this mitosis is done by eukaryotic cells, specifically somatic cells, so those are regular body cells. And it's not quite so simple as just copying and splitting because these cells are going to have multiple chromosomes and they have a nucleus, and there are additional steps that have to be taken care of to make sure you get two copies of that. Mitosis occurs anytime a eukaryotic organism would want to replace, repair, or grow and add more cells to itself. Last type of division that we will not talk about today, but we'll see in a future set of notes, is meiosis. And meiosis is also done by eukaryotic cells, but it's done to produce sex cells, so eggs and sperm. And a couple things that are different here: instead of 2 cells, you get 4 cells, but those 4 cells each have a half set of DNA. You might know that about egg and sperm cells. That's how fertilization works, because each cell has a half set of DNA. So when they combine in fertilization, the resulting zygote or embryo eventually has a full set of DNA. You can see here the original cell started with 4 chromosomes, and each of these 4 daughter cells has 2. Next is somatic cells versus germ cells. We mentioned this a couple weeks ago, but here's a little refresher. Somatic cells are basically your body cells, so everything that is not egg or sperm, so that's most of the cells in your body. Those are made by mitosis. Germ cells are egg and sperm. They're produced by meiosis. And like we said, each egg or sperm, each germ cell has a half set of DNA, which is how fertilization works. Remember that term germ cell, when we mean it here, is not like germs, like microbes that make you sick. It refers to eggs and sperm. Okay, DNA and chromosomes. We've talked a little bit about DNA as a molecule, but we haven't really talked about the different forms that molecule takes when it's in a cell. Usually, the DNA is in this uncoiled stringy format called chromatin, and that's because it's more accessible. It's kind of like spaghetti, like these long strands of DNA. That's so RNA polymerase can go along the DNA and make mRNA and do protein synthesis, but it's harder to work with. So there are other forms of DNA called chromosomes, where the spaghetti is sort of packaged up into a nice, clean package that's easier to move around and divide. We use that for cell division, but the DNA is not accessible. So it's harder to do protein synthesis when you have the DNA all coiled up. Kind of like yarn. Yarn is easier to access and look at all the different parts of it, I guess, when you have it uncoiled, but it's really hard to work with. So when you're moving it, transporting it, giving it to someone else, it's really helpful to have it bundled up in these nice little skeins or balls of yarn. How you'll see that in diagrams is just like this. When you see squiggles in a cell, either in a microscope image or in a diagram, those squiggles represent chromatin. When you see defined little lines here, that represents chromosomes. Here's another image all the way down to the individual strand of DNA. To make a chromosome, you wind that DNA around these proteins called histones, and then you wrap it into a coil, and then you coil the coil, and then you squiggle that up, and you have a chromosome. So when you see these little blobs that we represent on our diagrams we use to represent chromosomes, remember that all that is DNA that's coiled and coiled and coiled and coiled, like this picture that you can see here. Different types of organisms have different numbers of chromosomes. Just something to kind of remember. Oftentimes we'll talk about humans because we have 46 chromosomes. Bacteria and other prokaryotes only have 1. Fruit flies have 8. Oak trees have 24. Dogs have 78. Who knew that? That's kind of cool. Oftentimes our diagrams are going to be much simpler. We'll only show 2 or 4, maybe 6 or 8 chromosomes. We aren't going to draw all 46, but a human cell would be similar. There would just be more chromosomes, and that's harder to draw, harder to see. An important skill to have is counting chromosomes. So that's important because we want to make sure that each cell has the right number of chromosomes, and you want to be able to tell by looking at a cell which chromosomes are present and how many it has. The easiest way to do that is by counting centromeres. Centromeres are this region in the middle of the chromosome, and the total number of centromeres is equal to the total number of chromosomes. So on the left, this is 1 chromosome. On the right, this is still 1 chromosome, even though it looks like there are 2 pieces there. Those 2 pieces are called sister chromatids. They're just identical copies of the chromosome you started with. And because they're joined at the centromere, while they're joined at the centromere, we still count this as a single replicated chromosome. Here's another example. So these are unreplicated chromosomes. This is 2 chromosomes on the left. This is 2 chromosomes on the right. We call them homologous. That's just a fancy term for copies of the same chromosome. And if you count centromeres, you'll see that you only have 2 centromeres over here. And like I said, the best way to tell how many chromosomes you have is count the total number of centromeres. Here's some more pictures. I'll just count them with you. So here you can see 1, 2, 3, 4, 5, 6 chromosomes over here. Notice there are also 2 copies of each one, 2 bigs, 2 smalls, 2 mediums. While they're still bound together, we count each X or each pair of sister chromatids as 1. So 6 chromosomes on the left. And on the right, if we count centromeres, you have 1, 2, 3, 4, 5, 6, 7, 8 chromosomes on the right over here because those centromeres have split. All right, here's a little bit more about binary fission. Remember, this is how bacteria and prokaryotes divide. Really simple because you only have 1 chromosome. The DNA is copied and the cells split in half, and you have 2 identical copies because the DNA is a copy of the 1 piece that you started with. Simple, easy, kind of open and shut process. Now we're going to talk about mitosis, which is what happens in your cells and other eukaryotic cells, which is just a little bit more complicated because we have multiple chromosomes and a nucleus to deal with. So this is division of eukaryotic somatic cells. What we're going to end up with is 2 identical copies of the cell that we started with. Some people think of this as like a cell copy machine. You put a paper on a copy machine and press go. When you're done, you have 2 identical copies of the thing that you started with. And like I said, more complicated because now we have a nucleus and multiple chromosomes. And because we're humans and we like order, we break it down into these 4 stages. In reality, it's this continuous fluid process, but it's easier to understand what's happening if we give each stage a name. And all this is part of the cell cycle. This is just the M phase, that little wedge of the pie that comes after G2. So here's that whole cell cycle again. We are zooming in on this section here. These are the different steps of mitosis. Let's look at them in detail. So first is interphase. Interphase includes all of the stuff in blue over here. So most of the cell cycle, G1, S, and G2. If you want, there's little like word tricks, mnemonics here to remind you what the different phases are. So think interphase, interesting things happen during interphase. how interesting these things are, you can determine that. It's just a little thing that might help you remember it. But this is most of a cell's life. The cell performs its normal function, grows, and prepares to divide. The DNA is copied during the S phase, and at the end of interphase, we are ready to start mitosis. The steps of mitosis, you might remember PMAT, PMAT, prophase, metaphase, anaphase, telophase. That's how most people remember it, just with PMAT. So here's the first one. Prophase, think P for prophase, P for pair up. Chromosomes pair up. So our DNA started out as chromatin, like the stringy spaghetti. We're about to divide. We don't want spaghetti. Spaghetti's hard to work with. So the chromosomes condense. The DNA condenses into these nice chromosomes. Notice how there are 2 copies of each chromatid, and they are all condensed. Other things that happen during prophase, these organelles called centrioles move to opposite ends of the cells. So there's a pair of centrioles here, and the membrane around the nucleus dissolves. So remember, that nuclear membrane has been protecting the DNA while the cell is doing its functions, but now we want to start moving chromosomes around. We're going to have to get rid of the membrane. So you can see it breaking down here. And there's a picture of an actual cell. In prophase, you can see that chromosomes are condensed. Membrane is starting to break down, but we don't really see anything else yet. Next is metaphase. This is usually the easiest one to identify and the easy one to remember. Metaphase middle. Chromosomes line up in the middle. It's called the metaphase plate. There's not an actual plate. There's not a physical thing here that's holding these chromosomes in place, but it kind of looks like there is. So that's why it's called the metaphase plate. Also, the chromosomes will attach to these spindle fibers at the centromere, that middle portion of the chromosome. And then the spindle fibers connect to the centrioles at opposite ends of the cell. Here's a cell in metaphase. This is why it's usually pretty easy to identify, because you've got all these chromosomes in a straight line across the middle. Next is anaphase. A for anaphase, A for apart. This is usually a really brief phase, because after they're lined up in the middle, sister chromatids separate and pull apart to opposite ends of the cell. There's anaphase. You can clearly see 2 sets of chromosomes getting pulled to the top and bottom of that cell. We still have 1 cell until we are done with the last 2 phases, telophase and cytokinesis. Some people consider cytokinesis as a separate phase. Some people consider it part of telophase. It doesn't really matter. We'll talk about cytokinesis in a second, but if you think telophase, now we are starting to have 2 cells. So we're done moving chromosomes, so they decondense. They go back to the spaghetti chromatin format. These spindle fibers break down because we don't need them, and our nuclear membrane comes back. We have 2 nuclei, 1 around each of those cells. And then finally, we take the cytoplasm and split it. That is cytokinesis. There's telophase there. Cytokinesis means splitting the cell membrane. So you can see that once you take the cytoplasm, kinesis means cutting. So we are taking the membrane and splitting it, taking the cytoplasm and fully splitting it. So we have 2 separate cells now. That process is slightly different in plants and animals. In animals, the membrane just pinches and closes because we have this fluid membrane. Plants have a cell wall, so they can't just take that wall and sort of crunch it in. They grow a plate in the middle. It's called the cell plate that gradually connects the 2 cell walls together. So here are the different phases in a slice of plant material. So here you can see prophase, chromosomes starting to condense, nucleus starting to break down. Next comes metaphase. That's the easy one to identify. Then anaphase, they get pulled apart. Telophase, they're done dividing. They start to decondense. And then last is cytokinesis. So here's cytokinesis in animal cells. You can see the membrane starting to pinch in on both sides. And then here's cytokinesis in a plant cell. You can see this faint sort of cell plate forming between the two. When it grows all the way across, you'll have two plant cells there. Here are some more cells or more pictures showing cells in different parts of the cell mitosis process here. So this is probably interphase on the left. We can't really see individual chromosomes. Cell just kind of looks like a normal cell with a nucleus. Prophase can sometimes be hard to see, but it looks like chromosomes might be starting to condense. Usually in these images, you're not going to be able to see the spindle fibers or the centrioles, but they would be moving to opposite ends of the cell. There's metaphase, the chromosomes lining up in the middle. Anaphase, chromosomes getting pulled apart. Telophase, they've separated to opposite ends and they start to decondense. And then when cytokinesis is done, you can see we've got a wall between them and we have two separate cells. Here's some more pictures. I'd recommend open the presentation on your own and look at the pictures in detail, see if you can identify which is which. And here they are in an animal cell, pretty much the same thing except without the cell plate at the end. And I think that'll do it. If you have any questions, I will see you in office hours. Thanks for watching, and good job with mitosis. Your cells are doing it right now.